Good afternoon. Welcome to very special medical grand rounds. I'm Bob Wachter, chair of the department. Today, I think for most of you probably know, is a day we're celebrating the life and legacy of Floyd Rector, our late chief of nephrology and chair of the Department of Medicine. That event will begin here in this room at 2 o'clock, go from 2 till 5. After that will be the residents' annual Rector Research Symposium, where the residents showcase their research. That's at Faculty Alumni House starting at 5:30. Our guest speaker for the entire enterprise, not only this afternoon, but he spoke to the renal division this morning and to a genetics conference yesterday, so we're very much getting our money's worth, is Dr. Rick Lifton. His topic today couldn't be more relevant as we struggle to defend the importance of science and innovation and learning in the current environment, something we hold dear. And there are very few people in American medicine who've made more contributions in that regard than Rick has. He's president of the Rockefeller University. He's former chair of genetics at Yale and founded the Yale Center for Genome Analysis. He also serves on the board of directors at Roche and at Genentech. He's served on the advisory council to the NIH director, co-chaired the National Academy of Sciences and Royal Society's International Commission on the Use of Genome Editing, co-chaired the White House and NIH Precision Medicine Planning Group. He's also been awarded the Breakthrough Prize in Life Sciences and the Association of American Physicians Kober Medal. He graduated summa cum laude from Dartmouth and got his MD and PhD in biochemistry from Stanford, and then trained in internal medicine at the Brigham. He's received many, many honors, too many to list here, but has honorary doctorates from Northwestern, Mount Sinai, and Yale. And his talk today is aptly entitled "The Path from NIH Funding of Basic Sciences to New Therapeutics." And Rick, we're grateful to you for being here, and please welcome Rick Lifton.
Thank you so much, Bob. It's a real pleasure to be back to UCSF. It's one of my favorite places to visit for obvious reasons. These are my disclosures. And I just want to start relevant to the title of today's talk. I've used human genetics and genomics to try to understand human disease, and I just want to reflect for a moment on what would have been missed if NIH had not funded curiosity-driven research for the last 80 years. And just in my field, we would have missed that DNA is the genetic material, the genetic code, the discovery of recombinant DNA, discovery of methods to efficiently sequence DNA, the Human Genome Project, the discovery of CRISPR. All of this came from fundamental curiosity-driven research, almost all of it by NIH funding. And the revolution that we're in the midst of now in understanding the pathogenesis and hopefully the treatment of human disease never would have happened without all of this. And I know it's not lost on this audience what's at risk in the current discussions about the future of NIH and the funding of great institutions like UCSF. Since this is Medical Grand Rounds, I thought I'd begin with a case presentation. It's a 17-year-old boy who was transferred to Yale from an outside hospital with a history of 12 days of fevers around 39 degrees centigrade, watery diarrhea, developed a coagulopathy and progressive pancytopenia. He had elevated markers of systemic inflammation with his ferritin being elevated, and no infectious agent was identified. His course deteriorated, and he died from pulmonary hemorrhage on day 23 of his life. 4 days before death, his blood was drawn from him for exome sequencing. Surprisingly, as we collected his medical records from the outside hospital, 2 days after the funeral of the boy, his 43-year-old father presented with high fever, acute respiratory distress syndrome, subarachnoid hemorrhage, and hematochezia, and no infectious agent was identified. He developed DIC and pancytopenia, and he also had elevated levels of ferritin and ultimately markedly elevated levels of IL-18. Bone marrow biopsy showed erythro and myelophagocytosis, Ultrasound showed splenomegaly, and he had NK cell lymphopenia. He was treated with IVIG, dexamethasone, and cyclosporin, and he recovered 9 weeks later. Interestingly, in sequencing him and his son, we found that they had shared a previously unidentified variant in the gene NLRC4, which had not previously been implicated in disease. But the mutation was an interesting one, occurring at the ADP binding site that is exchanged with ATP. And it seemed like a potentially interesting mutation, particularly since this is a gene that is involved in an NLRC4-based inflammasome that responds to flagellin proteins when there's an infectious agent that gets into the cell, binds to NAPE, which binds to NLRC4, and kicks off the production of interleukin-1 and interleukin-18. It was intriguing that when we managed to recruit his parents, the father's parents, neither of them, who were the biological parents, had the mutation. When the adult recovered, he had a very interesting story to tell. He said, "I had considered myself to be generally healthy. However, it is true that when I was a child, I was hospitalized at Boston Children's Hospital for the first month of life, and every time they tried to feed me, I spiked a fever, developed terrible diarrhea, and I ended up being written up in the medical literature, and you can probably find the paper." He was one of the first 7 children in the United States treated with total parenteral nutrition. Amazingly, he had a rocky first year, but then recovered and had what he thought was normal life. However, he grew to an adult height of about 4 feet 10 inches, and he knew that he had a periodic fever syndrome that he just considered to be part of who he is, like we all have our own quirks. And he just considered this part of his normal life. And his characterization of it was that I knew that whenever I incurred emotional or physical stress, I would spike a fever, sometimes as high as 105 degrees Fahrenheit, but I'd get better in 2 or 3 days. I knew that I was going to get worse when my son died, and this is what ended up. So shown here is these are the ferritin and IL-18 levels. This is the ferritin level in the deceased son. This is the adult man who had very high levels that were suppressed with dexamethasone, IVIG, and cyclosporin. He has a son by a different mother. We contacted the mom and said, "How's your son?" "Oh, he's fine. Tell me about his history." "Well, he had this funky GI thing for about the first year of life, but he got over that, and he's done fine since then." "Do you have any fevers?" "Oh, fevers. Let me tell you about fevers. Every time he doesn't have a good night's sleep, I know he's going to spike a fever the next day." "Bad?" "No, not too bad. 103, 104, sometimes 105. So this was an incredible demonstration. And it turned out that sequencing the parents, this was a de novo mutation. So we have a de novo mutation in the gene that makes sense. The pathophysiology, certainly the inflammatory markers make sense. And there's a treatment for it. There are interleukin-1 inhibitors, IL-18 inhibitors that are, I believe, still in development, but the anacarin is available. And it turns out that there are now other patients like this. Folks at the NIH, we'd presented this at a meeting, and they came up afterward and said, "We've got a patient who has the same syndrome and has a mutation instead of being at, I forget the position, 4 amino acids away in the same part of the protein, and it was a de novo mutation as well." So that established the causal relationship of these mutations. I think this is just a wonderful example of a rare patient who you don't understand, who you can now figure out and sometimes find an approach to therapy. So genetics has been incredibly fascinating to me, and I think contributing for the last 40 years to the discovery of genes. And there have been 3 major phases of genes contributing to health and disease. The first of these was driven by the development of complete genetic maps of the human genome, so that if you had a trait that was segregating in a family in a Mendelian fashion, single gene cause, you could map the location of it and ultimately identify the gene, even in the absence of genome sequencing, of having the whole genome sequence. It was very arduous, but very doable. And we celebrated the development of this technology yesterday with the symposium for Ray White, who really was the person who developed the genetic maps to the level that anybody could use them. The second era is the development of common variants in common disease, and that's been driven by SNP genotyping. These are quite different. The odds ratio, or if you've got a disease genotype in a Mendelian trait, the odds of that compared to the general population is 40,000, 250,000 to 1, huge odds ratio. In contrast, the common variations that contribute to human disease account for more of the total variation in the trait in the population, but each individual SNP contributes somewhere between 10 and 20% increase in risk. So rather than 40,000 to 1, the odds ratio is 1.2 to 1. So a very small effect variation. And then in the last 15 years, we've had the ability to sequence all the genes in the genome or complete genomes. And this is a definitive approach to understanding the role of genetic variation in health and disease. So these are the kinds of pedigrees that we used to struggle to identify and collect all of the patients. And it was very successful in defining the pathogenesis of about 3,000 genes between 1987 and 2009. And the question was, is there anything left to do? And I'll get to that. This is shown here are the human chromosomes. And each dot on this is each color represents a disease, and the location on the map, on the chromosome map, that has been associated with a particular disease by genome-wide association of common variation across the genome. And there are literally millions of these associations. So we saw the possibility of using next generation DNA sequencing to sequence all of the genes in the genome. And this is the first clinical diagnosis and the first clinical application of this technology, where you make, and we did this in collaboration with a company at Moffitt, down at Moffitt Field, called Nimblegen, that we synthesized baits for all of the exons of all of the genes in the genome, use those to go fishing in patients' DNA, hybridize to these baits, purify them, and subject them to next generation sequencing. And the first demonstration was identification of a mutation in a gene that caused severe diarrheal syndrome in a newborn infant with a homozygous variant that had never been seen before in humans, at a position that was completely conserved from worms to humans, and enabling a definitive clinical diagnosis. So we've been focused quite strongly on alleles with large phenotypic effect, believing that these are really nature's gift to biology and therapeutics. They identify a causal link between genotype and a human trait, and they can identify genes and pathways that can be manipulated for health benefit, and they tell you the magnitude of the beneficial effects that you can get by modulating the activity of a target. So we started out working on hypertension. Coming out of my medical residency and training at the Brigham, cardiovascular disease was well attended to by Gene Brunwald, and I thought hypertension was an impressive problem in that there are 1 billion people affected, 10 million attributable deaths every year. And this is Arthur Guyton's working model for the regulation of blood pressure, which had left a mystery of how do we explain hypertension? It's a disease of the brain, the heart, the kidney, the adrenal gland, or the vasculature itself. And this is pretty hard to tell, but he had intuitions about this, but no, it was very hard to prove. So we set out to collect families from around the world who were segregating what appeared to be a single gene disorder that resulted in either severe hypertension or severely low blood pressure, with the idea that we would recognize the pathways that could be manipulated for health benefit. And over a period of about a decade, we identified the first, I think, 15 genes that impacted this trait. And they all converged on how the kidney handles salt. So this is my crude diagram of a nephron. And in the thick ascending limb, you can get loss of function mutations in salt reabsorption pathways that cause massive salt wasting and severe hypotension. In the DCT, there are mutations that cause both severe hypotension and severe hypertension. This is actually a relatively mild form of hypotension. In the distal convoluted tubule, sodium channels that cause severe hypertension called Little syndrome, or loss of function mutations in the same gene that cause severe hypotension. And I'll return to the adrenal gland in a moment. The most impressive part was that there were the same genes. You could get gain of function mutations that drive blood pressure to the high end, loss of function to the extreme low end. And then we had the ability with exome sequencing to start thinking about the remaining problems that we hadn't solved with in the Mendelian era. And one of these is aldosterone-producing adenomas. So these are tumors that constitutively make aldosterone, which tells the kidney to hang on to more salt and water and causes hypertension. And these tumors account for 5 to 10% of patients with severe hypertension. And we sequenced tumor normal pairs from these patients and found that more than 40% of these patients had one of two mutations in a potassium channel called KCNJ5. And we showed biochemically that these mutations occur right at the pore of the potassium channel that uniquely, selectively allows potassium to pass through the pore, but no other ions. Well, in the presence of these recurrent mutations in KCNJ5, this glycine was mutated to an arginine, and that allowed a very specific effect, which is sodium can now traverse this pore as well. And this is some of the electrophysiology showing this. And these two mutations that alter the pore are found in 40% of patients with these as somatic mutations. So these are obviously related to the cause. These are very benign tumors. On average, there are only four to seven missense altering mutations in these tumors altogether. And this is in the presence, these are the causal mutations that drive this. And the mechanism of this is pretty straightforward. This potassium channel normally mediates the repolarization of the cell after a depolarization event has occurred. But these mutant channels now allow sodium to come in at a dose that does not kill the cell, amazingly, but allows sodium entry to lead to chronic depolarization of the cell. And depolarization opens voltage-gated calcium channels at a sufficiently precise dose to allow this is the driver for cell proliferation and production of the steroid, the rate-limiting step in aldosterone biosynthesis. So this single mutation accounts for the formation of the tumor and the elaboration of aldosterone. So this turns out to be one of about half a dozen different genes that can be mutated to cause these aldosterone-producing adenomas. Others are in calcium channels, and a couple of other genes, including the sodium-potassium ATPase. And we can account for almost all of these tumors now. There's another mystery that remained after the Mendelian phase, and that was a disease featuring hypertension with hyperkalemia. And this was a perplexing disease. Most of the hypertensive genes that relate to aldosterone you develop hypokalemia and metabolic alkalosis because you're stimulating the epithelial sodium channel by aldosterone action. These patients are quite different. They have hypertension and hyperkalemia. And that was a mystery because the kidney sees aldosterone in two conditions. In volume depletion, you activate the renin-angiotensin system, aldosterone is produced, and that promotes salt reabsorption. In hyperkalemia, that's a direct secretagogue for aldosterone secretion, and this leads to potassium secretion. So how the kidney distinguishes between these two is a mystery. And the patients who had this phenotype suggested that they were stuck in a state in which they could only use aldosterone to promote salt reabsorption and not promote potassium secretion, accounting for the hypertension and hyperkalemia. So using exome sequencing, we ultimately identified a novel serine threonine kinase called WNK4 that is a driver of this disease. And there are mutations in 3 genes that cause this disease. One is the serine threonine kinase WNK4, and the other 2 are a ubiquitin ligase. Cullen-3 is a promiscuous partner of many different proteins that target specific proteins for ubiquitination by the Cullen-3 ubiquitin ligase. And ubiquitination leads to the degradation of the bound protein. So we discovered missense mutations in WNK4, mutations that cause missplicing, making an in-frame deletion of Cullen-3 as a second mutation, and then missense mutations in Kelch-like 3 or recessive loss of function mutations anywhere in the protein. But particularly interesting were the ones in the binding sites. And it turns out that the mutations in WNK4 are paired up with the sites that are mutated in Kelch-like 3 in a crystal structure where these critical sites are binding to one another. And so a long series of biochemical studies by Lynn Boyden and Shigeru Shibata, shown here, ultimately demonstrated that the WNK4 pathway is regulated by angiotensin II. And in volume depletion, that leads to phospholipase C and protein kinase C, which phosphorylates Kelch-like 3 that prevents WNK4 from binding to Kelch-like 3. Conversely, the point mutations in WNK4 prevent their binding to Kelch-like 3. And so higher levels of WNK4 go further to phosphorylate a protein called SPAC, which phosphorylates the sodium chloride cotransporter, promoting sodium chloride reabsorption. And by mechanisms that are not fully understood yet, inhibits potassium secretion. One way of interpreting this is the sodium chloride cotransporter in the distal convoluted tubule. If you activate it, you're preventing distal delivery of sodium distally, so that inhibits potassium secretion as a mechanism of the hypertension and the hyperkalemia. You're not delivering enough sodium distally to enable K secretion. So getting back to impact, many of these Mendelian forms of hypertension have specific treatments that were actually on the market. Such as hydrochlorothiazide is a corrective agent for this hypertension and hyperkalemia. Amiloride can ameliorate Little syndrome. Exogenous cortisol can ameliorate the mutations that cause constitutive secretion of aldosterone and so forth. So now the National Academy of Medicine has performed a study suggesting reduction in sodium intake. The FDA agrees, the CDC agrees that reducing salt intake would be beneficial. Lee Goldman and Bivens Domingo and Glenn Chertow had a paper suggesting the impact of a modest reduction in salt intake would be very important. The DASH diet, which also lowers blood pressure, Its major difference, only major difference really, is a markedly increased dietary intake of potassium. And getting back to the hypertension and hyperkalemia, if you eat a lot of potassium, you are obliged to secrete the potassium, but you do that at the expense of no longer being able to reabsorb sodium as well. So again, it enters into this final common pathway of reducing salt balance, lowers blood pressure. This led to a study in China that was quite imaginative. It's a 5-year cluster trial where they randomized not individuals, but villages. So 600 villages were randomized, matched and randomized to either normal salt diet or a 75% salt, 25% KCl diet. And this was run in 21,000 subjects with a prior stroke or were 60 years or older with hypertension. And they followed up and measured urinary electrolytes, found that people were more or less doing well with the diet. And over the course of the study, demonstrated that this lowered blood pressure, very modest effect of about 3 millimeters of mercury on average. But it had surprisingly impressive effects on reduction. This modest reduction in salt intake reduced the incidence of stroke, particularly hemorrhagic stroke was reduced by 30%, and all-cause death was reduced by 12%. And all of these are, because of the size of the study, significant at P001. So they're very good drugs for hypertension, but one of the big challenges is that people don't stay on the drugs very effectively. Average time on drug is between 30 and 90 days on average. And that's not very good for a drug that you should be on for the rest of your life. So one of the first studies that we did was to show that modest differences in angiotensinogen, which is made by the liver, are alter blood pressure because they alter the rate of formation of angiotensin II. And this resulted in a company called Alnylam making an siRNA against angiotensinogen. So short interfering RNAs get avid uptake by the liver, and they have an antisense strand that binds to the messenger RNA for a targeted RNA encoding protein and lead to its degradation. And there are drugs on the market that they've developed for transthyretin amyloidosis and others. And so they're going after angiotensinogen as a target where if you could knock it down and keep it down, this might be an excellent backbone for treatment of hypertension. So this is a normal level of angiotensinogen without treatment. And with a single dose of the siRNA for angiotensinogen, they get this spectacular knockdown of about 95% reduction at the highest dose that persists over a 6-month course. And this is associated with about 10 millimeter lower blood pressure, which is a good effect for any antihypertensive. And it's combinable with all of the other commonly used antihypertensives, in particular with diuretic, which adds another 12 millimeter reduction because of the synergy of having a diuretic in combination with an inhibitor of the renin-angiotensin system, which gets activated by the diuretic. So this is one example of new therapeutics where commonly asked in genetics, when are you going to stop finding genes and start having therapies? Well, this is one example. Here's another. So this is starts with an index case who was in a motor vehicle accident in Bridgeport, Connecticut. Is taken to the emergency room, had spines of his x-rays of his cervical spine. They were surprised at the apparent bone density, x-rayed other sites, looked like all of them had dense bones. And he was referred to Yale because they were concerned he might have multiple myeloma. So Carl Insonia, a bone specialist at Yale, saw him and said, yeah, let's just get his measurement of his bone density. And his Z-score in his spine was 8.1, 8 standard deviations removed from the mean of the population. This is the man with the highest bone density on the planet. So, but not by much. Half of his first-degree relatives also had Z-scores over 6, and the others had completely normal bone density. And the amazing thing about these patients is they had, aside from torus in the palate, their only medical complaint was that they sank when they tried to swim because they had such amazing bone density. So we thought we should try to figure out what the cause of this was, because this could be biologically interesting. And turned out that this is caused by mutations in the gene LRP5 that is a co-receptor in the Wnt signaling pathway, and is usually attenuated by binding to an inhibitor called DKK. And this leads to clearance from the cell surface and attenuation of Wnt signaling. So it's suggested that this was a critical pathway. These are different mutations that we found in subsequent families. And it suggested that you might be able to, if you could figure out how to mimic this inhibition, you might be able to have impact. So it turned out that there was a recessive disease that caused what was described as a sclerotic bone disease that was caused by recessive mutations in a gene called sclerostin because of the sclerosis. And Rob Krumloff showed that this protein was in fact the partner, the DKK-like partner. It was not recognized as being related to Wnt signaling until Rob put 2 and 2 together and showed that sclerostin is the inhibitor of LRP5, and these mutations that we identified prevent sclerostin from binding. So this led to the suggestion that you might be able to increase, not just attenuate bone loss in patients with osteoporosis, but actually increase bone density rather rapidly. And Amgen ultimately developed monoclonal antibodies to sclerostin and showed that indeed in clinical trials, these led to rapid increases in bone density, and you achieve a peak at around 12 months. This reduces hip fractures, and this is now on the market as a treatment for patients with severe bone density that literally came from these extremely rare patients. So another disorder, just giving an example of a genome-wide association study that was productive. Ali Gharavi, a fellow in the laboratory, assembled a cohort of patients with IgA nephropathy, which is a disease that leads to glomerulonephritis and progressive renal failure. It's a common cause of kidney failure, most common cause of kidney failure in people of Asian ancestry, although diabetes is starting to catch up there. One of the largest signals from the genome-wide association study turned out to be a deletion of complement factor H receptors. And this is a receptor in the complement cascade. And prior to this discovery, it was recognized that complement had something to do with IgA nephropathy, but it wasn't clear which pathway you would like to inhibit. Should it be the classical pathway or the alternate pathway? Well, this identified clearly that complement factor H is in the alternate pathway, and so if you're going to go after a therapy, you should go after something in the alternate complement pathway. And Novartis has developed a drug that they now have FDA approval for called Fabhalta, that they have shown at least markedly reduces proteinuria in this disease, and studies are ongoing to see what the clinical course related to renal failure would be. Just on genome-wide association, the other among many discoveries, the largest effect discovery that I think has great population impact and potential therapeutic impact is on focal segmental glomerulosclerosis in people of African ancestry. Martin Pollack at Harvard discovered common variants in ApoL1 and 2 independent common variants in ApoL1 that are rare in most of Africa, but were swept to very high frequency in Western Africa, suggesting that it was under positive selection. And the positive selection in Africa is likely going to be malaria, but it wasn't malaria, it was trypanosomiasis in this case. And but these are protective against a particular version of trypanosomiasis rhodesians, that ApoL1 is a trypanolytic factor that punches holes in the membrane of the trypanosome. And this version of ApoL1 is effective in targeting Rhodesians, whereas others are not. And this turns out to be protective in Africa, but when you get two of these mutant alleles in the United States, you're at markedly increased risk of developing kidney failure in your 50s when you have two mutant alleles. And this is one of the major drivers of the increased risk of kidney failure in all of the dialysis clinics here in the US. And this is an interesting target that is being pursued for new agents to try to mitigate the impact of these mutations. So once we got through this initial Mendelian disorder, we started thinking about what about these other 17,000 genes that we have, and 70% of them are conserved among the across the vertebrate lineage. So why haven't we found Mendelian traits underlying these other 17,000 genes? Well, we suspected that many of these might be caused by de novo mutations with impaired reproductive fitness that might include prenatal lethality or incomplete penetrance, where not everybody who gets the mutation gets the disease, or that there's variable expressivity so that you might not recognize everybody has the mutation as having the same disease. So over the last 15 years, we have participated in one of the NIH Centers for Mendelian Genomics. And to date, we've sequenced more than 40,000 exomes for this project across about 100 different phenotypes. And we've identified at least 400 genes with genome-wide significance, another 500 that are almost all likely to be correct. And many of these are genes that have never been recognized before as associated with human disease, while many others are associated with other diseases already, but different types of mutations cause different diseases. So I'll tell you about some of these. Here are some of the phenotypes that we've studied, and we've been, I'm a proselytizer by heart and have had a great time in my career of going to different clinical departments and saying, what's the most interesting problem that you don't know anything about the biology of, and establishing collaborations. And these include diseases like autism, where Matt State, who's now chair of psychiatry here, started his career in the lab, collecting families and doing first de novo mutation screens in autism that were successful. Structural brain abnormalities, another kidney disease, congenital hydrocephalus, intracranial hemorrhage, temporal lobe epilepsy, pediatric liver failure. And all of these were started by finding a smart fellow who was looking for something to do in their area and pursuing this project. So this is Marat Gunel, who started as a fellow in the lab when he was a neurosurgery resident. He's now chair of neurosurgery at Yale. One of his projects was looking for mutations that alter the development of the human neocortex. And he collected 1,000 patients from Turkey from consanguineous union with malformations of the brain. So you can see, compared to a normal 3-year-old, this MRI shows a brain that has none of the normal cortical folds, and this patient had the mutation in WDR62. was not recognized until you had the genotypes of multiple patients who had this mutation in this gene, that they actually shared very similar MRIs, because no physician had ever seen more than one of these. And so it was only retrospectively that you could categorize. It's kind of the phenotype second, just sequence everybody and see what falls out. And here's another example where normal cortical development posteriorly are caused by mutations in LAMC3. Mutations in NDE1 causes profound malformation of the brain. And he's found about 20 of these. Joe Gleason at UCSD has found another 20. And it's a remarkable collection of these disorders. Murat has also discovered the molecular basis of a common neurosurgical tumor, meningioma, benign tumors. This is Keith Choate, who was an MD PhD student in the lab who came back as a fellow wanting to do genetics. And we said, well, let's just find, and he had the intuition that you would find this patients would present themselves because they were going to have defects in the epithelium, and he's identified a bunch of these. I think the most interesting was the day that he came to the lab with these two photographs of patients that he had seen in the clinic with a disease called ichthyosis with confetti. And it turns out that the red skin is the ichthyosis, and the confetti are these white spots that they develop with age. They're not born with these spots. And so we were spitballing what might be the cause of this. And one guess was that maybe these are clonal revertants of an underlying mutation. This happens in Bloom syndrome, but had never been described in a skin disorder. And so we biopsied a couple of these spots to start with, and found that, in fact, the disease skin had retention of nuclei in the stratum corneum, very funny looking cells down below, and the revertant spots, these white spots, look just like normal skin. So these were clearly going to be revertants. And this was before exome sequencing. We had no good way to find the gene, but we genotyped a bunch of these spots and picked about 25 of them, and found that they all had somatic recombination that had made them homozygous for the 2 copies of the same chromosome in every case, which led us to keratin 10, which is one of the keratin proteins in the skin, and found that all these patients had frameshift mutations in the C-terminal part of the protein. And there's this remarkable glycine repeat that goes in the normal protein, and all of the mutations had frameshifts that into the same open reading frame that was loaded with arginines. And what happens with arginine? You now have this incredible positive charge. And these all end up being clustered in the nucleus rather than being in the cytoplasm anymore. And when they get these mitotic recombinants, it cures the disease in that local spot. Keith has gone on to do many of these, and he's now chair of genetics, chair of dermatology at Yale. This is Chris Colley, who was an MD PhD student in the lab, who's now chief of pediatric neurosurgery at Mass General. He got interested in congenital hydrocephalus, and this is a neurosurgical disease. These kids have massively large ventricles, and they get shunted. And the question is, do the shunts do any good? And nobody's really quite sure. And you ask, what's the pressure in this hydrocephalus? Nobody measures it. So, interesting. So Chris recruited a bunch of families by finding Facebook groups that were support groups, and almost overnight recruited about 200 families with kids with congenital hydrocephalus. And he has identified half a dozen mutations in these patients and different genes. And one of them is just interesting because it's TRIM71, which has it's an RNA binding protein that's involved with LET7, which is very famous for its role in neural development in C. elegans and also in flies and also in mice. These mutations alter arginines that are sites of direct contact with the cognate RNAs that they bind. It's expressed only in the periventricular zone in early development, and this is where neurons are born in the brain. It has led to the recognition that in these and many other of these patients, they don't have a problem with making too much fluid. They make too few neurons. So they have a primary neuronal defect. The shunting probably is not doing them any good except ensuring that they're going to have a lifelong shunt that's going to need revision every couple of years. Chris has also gone on to collaborate with Dennis Spencer, a neurosurgeon at Yale who does surgery for temporal lobe epilepsy. And by getting little bits of tissue that were taken out as the focus of the epilepsy, which turned out to be perihypothalamic in most of the cases, there are somatic mutations that are found when you do deep sequencing in these tissues compared to normal in the RAS-MAP kinase pathway, which came as a surprise to me. I thought, oh, well, you're just going to find ion channels, and others had found ion channel mutations in focal epilepsies. But these are all in mutations in genes in the RAS-MAP kinase pathway that we classically think of as being hyperproliferative mutations. It's unclear how they cause the epilepsy and whether they're actually replicating, but it's quite a convincing example. I mentioned incomplete penetrance. Chris Garcia, then at UT Southwestern, collected families with pulmonary fibrosis. Pulmonary fibrosis has been recognized to be a heritable disease featuring short telomeres that lead to death of pulmonary epithelium and resulting COPD at early ages. She collected families that did not have mutations in these genes. We sequenced a bunch of them and identified 2 novel genes that are implicated in pulmonary fibrosis. This shows one of the families, and there's a lot of incomplete penetrance. Not everybody who gets the mutation gets the disease, but it turns out that there's an environmental cofactor that plays an obvious role in this disease. Everybody who gets disease in these families, of which there are many that have been studied, among the affected, 60% are smokers. Others are chicken farmers, bird breeders, welders, masons, feed millers, farmers, cattle farmers, other construction workers. Perhaps you get the picture that inhalational exposure is the driver of whether people are going to get the phenotypic exposure or not. The ones who remain free of disease despite having the mutation work in an office, stay in the home, and do not have these inhalational exposures. It's a beautiful example of gene by environment interaction. And the last one I'll show, or maybe not quite, is de novo mutations that cause craniosynostosis. And this is Andrew Timberlake, who is an MD PhD student in the lab, interested in reconstructive surgery. Craniosynostosis is premature fusion of the bones of the skull. And the reason I mention it to you, it's one of the only good examples of a digenic phenotype. Andrew sequenced a bunch of patients with sagittal non-syndromic craniosynostosis and found that about 10% of them had loss of function mutations in the gene SMAD6. SMAD6 is interesting because it's in the bone morphogenetic protein pathway that promotes osteoblast differentiation and bone formation. And so heterozygotes for these loss of function mutations in SMAD6, which is an inhibitor of the pathway, would be expected to increase the activity of osteoblast differentiation, but not sufficient to cause the disease in many of the patients who transmitted this mutation to their affected offspring. Turns out that there's a common variant in BMP, the ligand that kicks off this pathway that increases activity of the pathway. And in these families, The people who get both the increased production of BMP and the loss of inhibition of SMAD6 accounts for the expression of the disease. So this is one of the few examples of a digenic phenotype. In the last minute or two, I'll tell you about a paper that we just published last week from the Heart, Lung, and Blood Pediatric Genomics Consortium. It's 11,500 kids who, with severe congenital heart disease who were sequenced. It was known for a long time that these kids are at high risk for extracardiac malformations, as well as neurodevelopmental defects, including autism. It wasn't known what determines those outcomes. So the sequencing showed that there are 60 genes that are mutated more often than expected by chance. And when you look at this list, there are some genes that cause a monotonic defect, conotruncal defect, tetralogy of Fallot, and they always give one phenotype and rarely give others. There are other genes that, when mutated, give you many different congenital heart phenotypes. Notch1, a classic developmental gene, can give you conotruncal disease, tetralogy of Fallot, left ventricular outflow tract defects, and hypoplastic left heart syndrome. The extracardiac manifestations, again, some genes never cause extracardiac manifestations. Others cause both extracardiac manifestations and neurodevelopmental abnormalities. And some genes are quite variable in the expression of these as well. I won't take you through the data on Notch1, which I've mentioned. There's one pathway that jumps out on this, and that's covalent chromatin modifiers, which are known to regulate gene expression. These are altering the amino termini of histone H3 or histone H4 predominantly, and regulate gene expression. And the most interesting outcome in this regard is sometimes the same genes in congenital heart disease are also significantly mutated in autism, such as POGZ. And in other cases, there are paralogs where the genes that do the same thing just turn out to be different in the heart and the brain. For example, the histone methylase at H3K4 and the reader of that mark, CHD7, the same loss of function mutations appear in autism, but they're in different genes that mediate those effects, MLL3 and CHD8 in the case of autism. And this turns out to be a common theme in a number of congenital malformations. If I have a couple minutes more, I'll just mention quickly that the major change in the last couple of years is the ability to treat diseases that we previously considered untreatable. Cystic fibrosis is a beautiful example, and the most reason I show it is it suggests that many recessive diseases may be correctable by chemicals that modify the folding of misfolded proteins. And this has been a change of life event for patients with cystic fibrosis. Similarly, the gene may not always be the target. PARP inhibitors, Alan Ashworth and others showed that if you have a BRCA1 mutation, you can get synthetic lethality by adding BRCA1 inhibits double-strand break repair by homologous recombination. PARP inhibitors inhibit the base excision repair pathway and cause synthetic lethality. Treatment of hemophilia has been transformed by a purely protein chemistry approach of making antibodies that supplant the need for factor VIII by finding pairs of antibodies that together bring factor IX and 10 together in a way that factor VIII normally would. And these two antibodies have completely transformed the lives of patients with hemophilia. And you may be familiar as well with the CRISPR-Cas9 approach to sickle cell disease and beta thalassemia. And base editing is now showing is being used as a direct agent in to treat patients with to lower LDL cholesterol and by knockout of PCSK9 and correcting alpha-1 antitrypsin deficiency. And there are more of these that are in the works. So in the interest of time, I'll stop there. I appreciate your attention. I think it's a spectacular time to be making progress, not just in the diagnosis, but also in the treatment of disease. And let's just hope we have an NIH that will continue to support it. Thank you very much.
Thank you, Rick. That was terrific and inspiring as you think about what's going on today. We'll go over for a few minutes, maybe take 5 minutes or so for questions or comments. I'll start with one. You made the sort of aside that everybody sort of pushes on genetics and genomics and says this has been promised that we'd find therapies. You made the point that really in the last 3 to 5 years, we've started to come up with therapies for some common and really important diseases. Why did it take so long? Because there were promissory notes in 1990 that precision medicine is here and it's going to transform medicine. It took longer than I think most people thought it might.
Yeah, I think it's a fair question. I think the biggest surprise is that gene therapy seemed the obvious treatment for all of these recessive diseases. We'll come up with gene therapies and we'll just give it to patients. It'll be one shot and done. Gene therapy has turned out to be much more challenging than I think any of us imagined it would. And we still are struggling. There are gene therapies that are now available. They're not regular. They're hard to get to every tissue. And these have not become the mainstay that I think people expected. Additionally, understanding the biology has become is very challenging, and biology moves much slower than gene discovery does. But I think the new technology is what matters. siRNA is an example. We now have half a dozen registered drugs using siRNA in the liver. We're still limited by can we direct it to the tissues that we want to go. I'll point out that in addition to gene therapy, seeing 2 of my immunology friends in the audience, the other was immune therapy for cancer. And that was promised for 40 years and it was a failure. And I think many people just gave up on it. And now we have remarkable therapies. So it's very hard to predict when. But I think we know that in the absence of understanding pathophysiology of disease, we're pretty hopeless at coming up with therapies. So I do think this is the most important start, and we shouldn't guarantee that we'll be able to treat every disease, but at least we'll understand them.
Question? Bill Seaman, Rick, thank you very much. In the first family that you presented with mutations in the inflammasome pathway, the father said he could predict when he would get this because he would be under stress. You have any idea what that pathway is?
No, we don't. We thought obviously this is cortisol, right? And Neil Romberg did a lot of work trying to figure out what the pathway was, and to my knowledge, this is as yet not understood. I thought it was just obvious that this is just going to be cortisol, but it turns out that there's no good evidence that that is the driver of the pathway. We've got brilliant immunologists in the pathway. Maybe in the audience, maybe they have the answer.
Any of the immunologists want to comment? All right. Any other questions or comments? Yeah.
I should just mention that everybody seems to have this feature of stress-induced.
I'm just making an editorial comment that it's so unfortunate that you're at this great pivotal moment of maybe great breakthroughs with an administration that is going to probably make it much more difficult than it needs to be.
Yeah. So I think we all are contemplating the impact of what's happening with the current administration and funding for science being so up in the air. It's great that we in New York and you in San Francisco are, the 2 great philanthropic cities in the United States. I know that it's not unlimited resources, and it will not begin to replace NIH funding. But we have to hope that some level of understanding of not just what it means for patients, but also what it means for the economy. There's no doubt that NIH support is a positive, makes a positive return to the U.S. economy every year. Millions of people employed as a consequence of it. It's not by mistake that the world's biotechnology industry is in the United States and not in the rest of the world. But we're at risk of giving it away for no good reason that I can discern.
Well, on that note, let us quit and hope. Thank you for inspiring us.